Okay.
Okay. On height. Click on height. Okay. Perfectly fine. Mamta ma'am, please
Yeah,
start.
yes,
Okay.
I shall take over.
Okay.
So, very good
My
afternoon,
slides
Dr.
are.
Yeah, your slides are very well visible. Dr. Pooja, welcome, Dr. Pooja. Good afternoon to one and all. So, it is a matter of great pleasure to me to introduce our speaker for this session, Dr. Pooja Tamboli, who is an established animal researcher. Dr. Pooja Tamboli qualified JRF for the master's degree program and completed her MVSc in the discipline of livestock production management from ICAR NDRI Institute located in Haryana, Karnal. She did her PhD from the same institution, completed in 2019. Dr. Tamboli started her career as an ARS scientist in year 2020 at Indian Grassland and Fodder Research Institute, Jhansi. She is involved in research, extension, and teaching activities in the field of livestock production management since 2020. Dr. Tamboli is currently working on a project titled Foraging Behavior of Small Ruminants Under Natural Grassland in Bundelkhand region. She is also involved in other projects such as AICRP on goat improvement, feeding and management strategies to improve goat productivity under semi-arid climate, and soil total mix ration for livestock production. She is a member of many recognized societies and she has been awarded many prestigious awards like Best Scientist Award during Second Veterinary and Animal Science Congress. She contributed to the publication of 24 research papers, 23 technical articles, 11 book chapters, 2 books, 4 manuals, and 3 lead papers and 1 bulletin. She has presented 17 papers. So currently, she has played a role, significant role in organizing many training come workshops on different elements of animal rearing method, working as in charge fodder unit of IGFRI. So for today, the topic of our presentation is going to be modern technical trends in the science of silage making, and surely it will enlighten all the participants. So over to you, Dr. Pooja.
Okay. Um, Thank you, ma'am. Um, uh, I'm here to present about the modern technical trends in the science of silage making. Um, am I audible to all of you?
Yes, ma'am, you're audible.
Okay. So, what is the need? First thing is that we preserve the green fodder in the form of either hay, silage, or haylage, or anything. So, what is the need? So, there is need because during the rainy season, we have plenty of green fodders available. But in case of lean period in extreme summer or in the month of October, November, there may be deficiency of the fodder for the livestock. So keeping these things in mind, we can preserve the fodders when it is available in plenty in the form of silage or hay to meet demand of animals during the lean period. So it is just like achar which we prepare in our home. So when the season, there is no season of the mango, then we can also enjoy the pickles like that. So and there are three types of conservation methods. Green fodders can be preserved in the form of silage. Dry fodders, mostly the moisture content is around 14 or 15% in case of hay, and in between that there is also a type of preservation method, that is haylage. So one by one I will explain. Mainly I will focus in my lecture about the silage preparation. So what is the silage? Silage is a product of fermentation under the anaerobic condition in the structure called silo. in case of silage,
PPT तो नहीं हो
the
रहा है
dry matter is maintained around 30 to 35%, and it is preserved in green moist conditions. The color looks like a golden brown color after the preparation. And what is the goal? Main goal of making silage is actively growing wet plant material. If we keep this plant material as such in any storage condition, then there may be spoilage, maybe of fungal infection or anything else, because the form is unstable. So we convert it into the stable condition while preserving its forage nutrients
मैम
to feed
प्लीज
the animal in
मैम
later stage.
मैम पीपीटी शो नहीं हो रहा है
Okay. Visible है? My slides are visible or not?
Earlier it was visible, ma'am. Now it is not visible.
Now it is not visible.
Earlier it was visible. Who asked not visible? Actually, it was visible. Now it is visible.
Okay, again I am saying.
It is visible, ma'am.
Visible?
Yes. No, it is not visible.
Not visible.
Earlier it came.
Is it visible? Slides are visible.
Ma'am, screen is visible, but presentation. Yes, ma'am. Presentation is visible. now.
Yes. Yes. Now it's
Yes,
okay.
ma'am.
Okay. So I explained these things, and now just the unstable form of green fodder is converted into the stable form in the form of silage, while preserving their nutrient content, so that we can feed the animals at a later date. Now these are the steps of the silage preparation. First thing is that we should keep the dry matter content. The dry matter of the forage for the preparation of the silage should be around 30 to 35%. We should keep in mind their buffering capacity, carbohydrate content should be proper, and plant structure, means stage of harvesting, and weather condition. Means we can't prepare the silage in case of heavy rainfall or in extreme winter conditions. So the weather condition is also should be weather conditions should also be favorable when we are thinking for making the silage. And there are different types of methods for the preparation of silage, like in the trench, silo. Means many structures, many type of technologies are there for the preparation of silage: trench, silo, above ground, below ground. I will explain later on. Method of harvesting is also it depends upon method of harvesting. Air control is required. The condition while preparing silage at that time, the condition should be anaerobic. We can also add additives if required. If there is so many methods of additives like molasses, urea, antifungal agents, enzymes. So these are the additives. And also the compaction, means proper covering of the silo also is very important to create the anaerobic conditions. So these all it improves when the condition is proper for making the silage, then it will enhance feeding value and minimize nutrient losses. So what is ensiling? So ensiling, as I also described earlier, it is the preservation of green fodder under the anaerobic condition while maintaining its moisture content around 60 to 65%. So the objective is here there is an anaerobic fermentation in which the plant sugar, which are the soluble sugars, that convert into the acids, maybe lactic acid. Major portion is lactic acid, and up to the some extent CO2, ethanol, ammonia. And also in case of if the based on the substrate, some part also convert into the acetic acid. Next thing is that during the fermentation, there is also decrease of the activity of undesirable microorganisms like Clostridia or Enterobacteria, so that the nutrient losses can be minimized. So these are the different types of silo. Silo is the structure where the silage is prepared. So first is the below ground trench, above ground trench, below ground pits, above ground tower. And another methods are also like kacha silo, in simply there is no plastering, plastic drum silo, plastic bag silo. So we can prefer the methods based on the conditions, area we have. If sufficient area is available, then we can mostly prepare that pit silo. If we have less space, then we can go for the bag silo, drum silo, or in case of the like in waterlogging condition, or in the area where there is heavy rainfall, then we can go towards the bag silo or drum silo. 1 minute. हेलो, थोड़ा सा मैं बाद में बात करूंगी. एक्चुअली मैं उसमें लेक्चर नहीं दी. So now we will see the what is the amount and size of the silo, and that if the chaffing material compacted properly, we have to first we should keep in mind the stage of harvesting. Then second thing is that chaffing of the material is required, and third thing is that the size of silo pit and the amount of how much amount can be kept in particular size of silo. So for every cubic meter silo, we can make 300 to 350 kg silage. So for one animal, if we provide 10 kg silage per day, then 300 kg can be prepared in a 1 cubic meter silo, can be fed for 1 month. So like that, accordingly, we can increase the size of the silo based on the number of animals and based on the time period for up to which we can we should feed to the animals. So based on the requirement, time, duration, and number of animals, we can increase the size of silo and the amount of silage which need to be prepared. So now another things are that crops suitable for silage making. So all the crops are not suitable for silage making. The best crops which is suitable for silage making are maize, sorghum, bajra, as they are rich in soluble sugar or carbohydrates. The cultivated natural grasses can be natural grasses like bajra napier hybrids. These are the some grasses, fodder sugarcane grasses. They can also be ensiled, but there is need of addition of molasses too at the rate of 3 to 3.5%. Molasses is needed when the amount of sugar or carbohydrate is less in case of silage. And if we have, it also depends upon every time we are not having the maize, sorghum, bajra. If we are having grasses, cereal fodder, legumes, in that condition also we can prepare the silage, but the ratio should be 3:1. In case of unwilted leguminous leafy fodder and dry forage, if we are having, then the ratio of the leguminous fodder and dry fodder should be at the rate of 4:1. So accordingly, we can prepare the silage based on the availability of fodder available with us. Now the second thing is that stage of harvesting. So we should keep in mind the harvesting stage. Mostly all the crops are harvested between flowering and milky stage when we are going for the preparation of silage. And as the thick stems are mostly conserved in the form of silage, while the thin stemmed crops are mostly conserved in the form of hay, like berseem, lucerne. They are also rich in protein, so they are conserved in the form of hay. But thick stem like jowar, bajra, maize, they are suitable for the preparation of silage. Now another important thing is that chaffing. Chaffing should be proper because for the preparation of the silage, the condition should be anaerobic. So if we are chaffing the fodder, then there will not be any air pockets. And also second thing is that compaction. So first thing is chaffing, then compaction and filling. While filling, the compaction should be proper in the form of trampling or or with the help of that tractor, like shown in case of figures. So we have to press properly so that the conditions should be anaerobic and there should not be any air pockets. And after the harvesting, chaffing and filling, proper compaction is needed, and then after compaction, proper covering is needed with the help of plastic sheet, mud plastering, or dung, or grasses, or anything else. A main thing is that we should avoid the entrance of air. So as I already described, that covering should be proper, because we all know the lactic acid bacteria will only work under the anaerobic condition, and the production of lactic acid is required for the preservation of the fodder in the form of silage. So around 40 to 60 days, we can get a good quality silage. Now, different phases of the silage preparation when we have covered. So initially, first thing is that we should decrease the filling time of silage. Means that minimum duration should be there for the filling of the silo pit in order to minimize the exposure to the airs. Although that if we have filled the silo pit, then there is some part is starting in initial phase. There is the aerobic condition. So first phase comes is aerobic phase, where plant enzyme and aerobic microorganisms are more active. So initially, after the covering, there is aerobic conditions. So in that case, when aerobic microorganisms are active, there may be losses in the form of heat, that nutrient lost, and also in the form of carbon dioxide. In that condition, temperature may increase up to 15 to 20 degree Fahrenheit. And as the condition progresses, then second phase comes, that is fermentation phase. In the fermentation phase, oxygen is completely exhausted. And then when oxygen is exhausted, the condition becomes anaerobic, and then anaerobic microorganisms like lactic acid bacteria starts growing, and they convert the water soluble carbohydrate into lactic acid, acetic acid, carbon dioxide, and ethanol. In this phase, the pH is around 5. The next third phase comes, that is stable phase. In stable phase, the silo pH is around 4 to 4.5. In this phase, the matter is that forage material is almost stable, because there is production of more amount of lactic acid, and the microbial, other type of undesirable microbial activities are minimal. Only those organisms which can be survived in case of acidic medium, they only grow, like lactic acid bacteria. Then fourth phase is feed out phase. Feed out phase is the phase when we are not following the proper condition of the silage preparation, like harvesting, filling, time duration, and all these things, and type of forage. So if the silage conditions are not proper, then, and also compaction is not proper, then environmental exposure may lead to fungal growth in the silage, and that is called feed out phase. And the silage prepared is not of good quality, and it cannot be fed to the animals. Now, based on the different fermentation pathway, there is production of different types of products in the silage. So the end products like lactate, acetate, ethanol. So these are the different type of end products, and based on the fermentation pathway and substrate, the products often differs. So the most important is that lactate dehydrogenase is very good enzyme which is responsible for the production of lactic acid. So the first phase is very good, because in that case we will obtain very good quality silage, and accordingly when the fermentation pathway goes towards pyruvate dehydrogenase pathway, then there is production of ethanol and CO2. In that case, there is no production of lactic acid, and the silage quality is not good. So based on the fermentation pathway, also the product often differs. Now, as I previously also I explained that for the preparation of silage, the water soluble carbohydrate should be more. In that condition, the homo fermentation occurs. In this homo fermentation, the lactic acid bacteria grows, and it convert the soluble sugars into the lactic acid with the help of lactate dehydrogenase enzyme. However, when the carbohydrate concentration is very low, in that condition, the heterofermentation occurs, and there is activity of Enterobacter as well as Clostridia, and there is less production of lactic acid and more production of another type of acids like acetate, butyric acid. So the putrefying smell also comes when the amount of protein is more in case of fodder. So the water soluble carbohydrate concentration should be high. Now, the pathway, mostly two types of pathway, fermentation types are there, that is homofermentative and heterofermentative. Homofermentative is good for the preparation of silage. In this case, the soluble sugars converts into the lactic acid with the help of lactic acid bacteria. But in case of heterofermentation, there is growth of different another types of microorganisms when the soluble sugars content is less in case of the fodder to be inside, like Enterobacter, Clostridia. So these organisms produces acetic acid, ethanol, carbon dioxide, and also the pH will increase in case of heterofermentation, and there is foul smell, putrefying smell will come from the silage, which is not favored by the animals. Now, these are the different types of pathways. Accordingly, we have assessed the recovery of energy. So, in case of homofermentation, the lactic acid bacteria is more active, and they convert 2 moles of lactate. While in case of heterofermentative, the bacteria produces 1 acetate and 1 lactate. And when we further go downward, then in another case like yeast, Clostridia, Enterobacteria, these are also microorganisms which results into heterofermentation, and the product obtained is not good, and there is less recovery of energy and dry matter. The best one is that in case of first phase, that is homofermentative, is very good for the preparation of silage, because in this case the recovery of energy is around 96.9%, and dry matter will is 100%, means there is no loss of any nutrient. But when heterofermentation occurs, then there is growth of many types of another undesirable microbes, which results into loss of energy as well as dry matter. So we should keep the all the conditions which is suitable for preparation of the silage. Now, the different factors which affect ensiling process, that is anaerobic condition, chaff length, compaction, covering. These all affect the preparation of silage. What amount of water soluble carbohydrate silage preparation also depends upon the soluble sugars like glucose, fructose, sucrose, fractans. Dry matter content should be proper. Lactobacillus bacteria, number of bacteria, type of bacteria. So these are all factors which affect the silage. Now the first thing is that anaerobic condition. Anaerobic condition is prerequisite for the preparation of silage. The silo pit in which we are preparing the silage should be anaerobic. Various factors such as filling, chaff length, and compaction, they are all affect preparation of the silage, as I already told earlier. And also in a research, it was observed that the filling time also affect the type of silage prepared. So the rapid filling of silo maximizes the silage quality because there is less exposure to the air. Because our main objective is the condition should be anaerobic. In a research, it was shown that the number of days when they reduced from 11 to 5 days for the filling of the silo pit, then there was decrease in the acid detergent insoluble nitrogen by 5.3%. Means the fiber bound nitrogen decreased when the time of filling was reduced. Size of chaff size, means before the filling, after harvesting, there is chaffing is required. So chaffing, the size of chaff length should be 0.75 to 1 inches. And another important thing is that density of packing is also important. Density should be around 300 kg per meter cube for proper fermentation of the material, so that the air pockets can be removed and proper anaerobic condition can be maintained. Now, the dry matter content of around 20% is unsuitable because there is the moisture content is more, and the more losses of the energy may occur in this case. So for proper fermentation, the dry matter should be around 30 to 35%. The dry matter of around 40 to 45% can be ensiled. But what happens if the actually for the silage making, we prefer carbohydrate rich fodder. But in case of protein forages, we can ensile the material when the dry matter content is around 40 to 45%. In that case, the proteolytic activity will be decreased. Otherwise, if the dry matter content is less, in that condition, the fermentation activity will be faster, and in presence of the high rate of enzymatic activity, there will be proteolytic activity, and production of putrefying smell will be there in case of protein rich fodder if we are using for the silage making. So if we are using protein rich forage, then the dry matter should be slightly higher. And in a research, it was reported that ensiling may be also possible when the dry matter is more, that is 60%. If the dry matter is 60%, also the matter can be ensiled. But what will happen? The silage will not be of good quality because of the less rate of fermentation. there is higher pH, means the lactic acid concentration will be lower and the pH will be higher. So the quality of silage is not good. Very good type silage is have pH of around 3.8 to 4.2. And if the pH of silage is more than 5, then the silage which we are preparing is not of good quality and not favored by the animals. Now, these are the different types of crops which are suitable for making of the silage, like maize, sorghum, oat, grasses. We should harvest the maize during 50% flowering to dove stage, and sorghum 50% flowering to dove stage. Oat should be harvested during wood to dove stage, and grasses during early flowering stage. Now the water-soluble content at the time of ensiling, it also plays major role. For the proper initiation of fermentation, around 6 to 8% water-soluble carbohydrate should be there at the time of ensiling. And also the water-soluble content of around 12 to 20% is very good for as the pH range of 3.8 to 4.2 will be obtained in the silage, and it is of very good quality silage. So if the water soluble content is 12 to 20%, then it is very much perfect for the preparation of silage. So we should also keep in mind about the soluble sugars. Now the different types of forages and water soluble content of the forage. And as we all know that the maize, oats are very good type of forage which are suitable for the preparation of silage. And these are the different content, like in case of maize, 22%, oats 16%, sorghum 8%, millet 7%, and in case of barsim, 5%, lucerne 4%, range grasses 2 to 4, and tree leaves 1 to 3%, their amount of water soluble carbohydrate. These are the constituents of non-structural carbohydrate, that is water soluble carbohydrate of different types of crops. In case of perennial ryegrass, the total amount of water soluble carbohydrate ranges 60 to 250 gram per kg dry matter. And in case of different parts, if we will see the glucose, fructose, sucrose, like 10 to 30 gram per kg dry matter is there in case of ryegrass. Fructose around 10 to 30%, sucrose 50 to 70%. Like that, these are the different amount of the different type, means in different type of crops, the amount of water soluble carbohydrate varies. Now the lactobacillus bacteria, there are different types of lactobacillus bacteria. As I already explained earlier, that type of lactobacillus bacteria also affect the quality of the silage. So if the lactobacillus plantarum is growing under the anaerobic condition, it has a vigorous growth rate and it is acid tolerance and has capacity to produce high amount of lactic acid. And in some another condition like lactic acid bacteria, like that lactobacillus acidophilus and lactobacillus bulgaricus, they can also be used in the form of microbial inoculants in combination with lactobacillus plantarum. The combination has high acid tolerance and they have faster growth, and there is production of more amount of lactic acid, which is required for the preservation of forage. ChemLac HD is a product, a microbial inoculant having combination of lactobacillus plantarum, lactobacillus acidophilus, lactobacillus bulgaricus, which contain around 136 billion colony forming unit per gram, and it can be mixed at the rate of 1 gram per ton to the chaffed fodder, which is to be ensiled. So microbial inoculant can also be added while preparation of the silage. Lactobacillus plantarum, Lactobacillus acidophilus, and Lactobacillus bulgaricus have the ability to dominate the fermentation, and they produce more amount of lactic acid and inhibit the growth of yeast and yeast mold and other fungi. Now, the enzymes can also be used as additives while preparation of the silage. Cellulase and hemicellulase have the most widely used enzyme complexes because they increases the amount of fermentable sugar concentrations. So we can spray this type of enzymes on the ensiling materials, which results into the increased release of residual sugars, and thus the silage preservations can be improved. And also, when we obtain good quality silage, the dry matter intake and production of animals will enhance. Thank you. And in additives, we can add another things also, like antioxidants can also be added, like BHA, BHT, butylated hydroxyanisole, butylated hydroxytoluene. These are all we can add. Antifungal agents like sodium benzoate, potassium sorbate. These are all another type of additives. So in order to improve the quality of silage, we can add these types of antifungal agents. Enzymes I already described. We can also add urea at the rate of 0.5%. Urea addition should be very less. Urea is added to increase the protein availability to the animals, but the amount should be less. Molasses can be added, but the rate should be around 1 to 3%. So these are some extra additives which can be added when we are going for the preparation of the silage. So these are all about the silage preparation. Now, if anyone have any doubt, they can clear with me, or they can ask.
Participants, if you have any questions, please ask the expert. Any questions?
जी मैम कांटेक्ट नंबर दे दीजिएगा संभव हो
Contact number I will type in chat
You
box.
will send mail to ma'am. It's not possible that she will share contact number with all participants. You can ask her questions here, or you can mail her questions. Ma'am, am I right?
Okay. My email ID is also given in here. You can note. My email ID is, you can